Hello, everybody, and welcome to our SpectroMine 3 launch seminar. My name is Max Helf from the Biognosys R&D team, and I welcome you here to a seminar on unearthing the full potential of DDA Proteomics with SpectroMine 3. To give you a brief overview of what we have planned for you today, we will start with an overview of the company and our software products, and then I will hand over to the lead developer of SpectroMine, Tikira Teemu, for an introduction to SpectroMine in general and the improvements that have been made in version 3. And finally, we have our guest speaker, Eivind Schulp from the Technical University of Denmark, to share with us how he uses SpectroMine for single-cell proteomics. And to get us started, as I said, I'm giving a brief introduction to the company. So Biognosys has been around for over 13 years now as a proteomics innovator and started as a spin-off from the lab of Rudi Ebersold at the ETH Zurich. We have a highly qualified workforce and our products are used widely in scientific literature. We also run one of the largest facilities in the world for large-scale discovery proteomics with our services department. We have customers all around the world, and we serve them, among other things, with our main software products, which include Spectronaut, which is our best-known software for data-independent acquisition. But we also have great products for targeted proteomics in SpectroDive and for data-dependent acquisition in SpectroMine. And that, of course, is the software you'll hear a lot more about today. So in general, our software products provide great performance. They're used very widely by the scientific community. And one of our strengths is that software is really easy to use. And this is something we continuously improve in collaboration with our customers. And we're really taking into account any feedback we get from our users. So for SpectroMine, it's really characterized by very strong identification performance, and especially for version 3, very high search speed. And one of the key features is its deep learning augmented search algorithms. It is specially designed for isobaric labeling quantification data analysis. We have a library-based DDA search workflow that is also extensively used, for instance, by Erwin. And there's full support for eye mobility technologies. And before I hand over to Tikira, I have a polling question for you, just so we can get to know the audience a little better. And the question is, have you used SpectroMine before? Yes or no? And I will just wait a little bit to see the answers come in before I continue. And with that, let's see the results. So yeah, a lot of potential new users here, but some of you have used the software before. So we hope that we can introduce you and get you excited about SpectroMine. So with that, I will hand over to the lead developer of SpectroMine, Tikira Teemu, and she will give you an introduction to the software in general and specifically what's new in SpectroMine 3.
Thank you, Max. Thank you very much for the lovely introduction. And also hello, everyone from my side to today's launch seminar about SpectroMine 3. So within the next 20 minutes, I will give you a brief introduction into Pulsar, our search engine. I will show you the DDA search workflows that we support, also what that means in terms of search space complexity, and then also conceptually how we do scoring so we can distinguish true from false identifications. Afterwards, we will dive a bit more into SpectroMine 3. I will show you the experiments that we support with the software. We will have a look at identification as well as quantification accuracy. And then last but not least, have a look at the user interface that you know what you're working with. So let's get started with Pulsar. Pulsar is our proprietary search engine. It was originally created for our in-house library generation pipeline for Spectronaut, but now it's very powerful and we use it in all of our software products. During the development, we focus on three main things. One is ID performance, second is also speed, and third is that it is able to handle large data sets. And here we mean both in terms of the amount of raw files it can process, but also on the complexity of the data set. So it supports analyzing experiments with many modifications. Let's have first a look at the classical Fasta search pipeline. So here we have biological samples, which are then measured on a mass analyzer. And we support 5 different vendors for that. In the end, we will have the DDA runs. In the classical Fasta search pipeline, we use a Fasta file, put that together with the DDA runs into Pulsar, and in a spectrum-centric search, we will retrieve our search results. In contrast to that, we have the library search pipeline. Here, obviously, it's the same biological samples that are measured on an analyzer, and then we have the DDA runs. But now we have 2 steps. In the first step, we use a Fasta file, and it's the same spectrum-centric search. We can add additional runs to create a library. The second step then is to use that library and search against that in a peptide-centric approach and to retrieve our search results. Now let's have a look what that means in terms of search space complexity. On the left-hand side, you can see the Fasta file. There we don't need much prior information. We can just download the Fasta file, for instance, from UniProt. But then we need the digest settings and the modifications. Therefore, the search space can become quite large until we come to our theoretical peptides. In contrast to that, on the right-hand side, we can see the library. There we need much more prior information since we can, for instance, include multiple DDA runs, but then the search space is much smaller and therefore the runs are faster and the ID performance is the same. Let's have a look at how we apply scoring in Pulsar. So what you can see here on the left-hand side is that we have PSMs, both for target and for decoy, and we use them equally throughout the whole process. The PSMs have multiple scores that describe characteristics of scans or peptides of the PSM, and the values can be discrete, continuous, or Boolean, for instance, just true or false. So in the first step, what we do is that we split the PSMs into n different splits. And for each of these splits, we create a classifier, a support vector machine. Afterwards, for each classifier, we use a different set, and then we score and predict them and classify them using the previously created classifier to end up at a machine learning score. Since we used multiple classifiers and not just one, we need to normalize that score until we can, in the end, apply FDR calculation. We tightly control FDR at different levels, at PSM, peptide, and protein group level, and we also thoroughly evaluate and validate this. Since we cannot go into all of that scores, I still want to highlight 2 scores in particular, and these are our deep learning scores. For that, we created a neural network with a multitude of data that covers different species, different cell types. We have also different proteases, phospho enrichment, ILQ, instruments, and a collision energy. And we have 2 scores that we predict out of that. So we have the delta IRT score that we use, and we have the spectral angle score on the right-hand side as well. The delta IRT score is the difference between the observed IRT and the predicted IRT out of that neural network. And the spectral angle score is basically an observed spectrum and then also the predicted spectrum to calculate the spectral angle score. And both of these scores help tremendously in discriminating true from false identification. And with that part, we're done with the insights into Pulsar. And before we go into which experiment SpectroMine 3 supports, we have another polling question where we would like to know, exactly, there it is, where we would like to know what you use quantitative DDA proteomics for. And it can also be 2 options, right? So we have label-free quantification, We have isobaric labeling quantification, or maybe you don't do DDA quantification at all. Okay, so I think we can have a look at the results. Great, so most of you do LFQ, then it's 71%, and 42 also do ILQ, and 23% don't do DDA quantification at all. So let's have a look what SpectroMine supports. So we have, first of all, isobaric labeling quantification that we support. And here, like most of you know, but still, I think it's good to remind all of us. So isobaric labels are a chemical group that are nominally identical in mass, but they vary in terms of their heavy isotopes that are distributed among the structure. Therefore, we have reporter ions that can tell us in the end which peptide and which sample is found how often, so it can be quantified. Looking at the concept here, so we have n different samples. Each of the sample is labeled differently. They're pooled together and then analyzed in one MS run. And so the beauty of it is that you just need one run, but still can quantify a multitude of samples. And examples for that are TMT, iTrack, or EasyTag. The second experiment that we support is the labeling efficiency check experiment. And there we use the same isotopic labels, but now the difference is that we're not pooling the samples together anymore. So we still have n different samples. Each of the sample is labeled with one reporter ion, but then they're analyzed differently. And the goal here is to figure out the percentage of labeled data. So we want to see in the end how good the labeling works. And this is usually done prior to any ILQ experiment, so you can get a feeling for the outcome. In contrast to that, to ILQ and to labeled samples, we also have label-free quantification. Here, as the name already tells us, there are no labels required. The samples are analyzed separately. But due to the fact that DDA is semi-stochastic, we cannot rely on MS2 information because some precursor ions might just not be used for fragmentation. Therefore, for LFQ precursor quantification, we use the precursors MS1 peak. And there is an important setting that we have in SpectroMine. So we have the experiment settings, and there's a pre-configured schema with label-free. And the important part is, as you can see down here, is that the XIC construction has to be switched on. So to summarize that, what we support in SpectroMine 3 are mainly ILQ experiments. So we have a lot of pre-configured examples here for you. So we have TMT as well as TMT Pro, so all the latest TMT labeling. We have iTrack and EasyTag. But in theory, you can configure whatever you like, so any ILQ experiment is supported. We have the same pre-configuration for labeling check efficiency, and then we also support label-free quantification, library generation, and all the experiments, as I've shown you in the beginning with Pulsar, can be searched against a Fasta file or a library. Next up, it's also quite important that we see how SpectroMine performs in terms of identification and also for the quantification accuracy. Therefore, I first have another polling question for you. So which software are you using to analyze your DDA data? And sometimes it's not just one, so it can also be a multi-selection here. SpectroMine, MaxQuant, Proteome Discoverer, Peaks, or another software. Okay, I think we're done, and let's have a look at the outcome. Since most of you haven't heard of SpectroMine so far, it's not surprising that not many of you use that currently to analyze your DDA data, but maybe after the next few slides, I can convince you that it's worth a try. So let's first have a look at how SpectroMine performs in terms of identification. So we compared SpectroMine to Peaks, Proteome Discoverer, and MaxQuant, and we used 9 different data sets for that. 7 of these datasets are ILQ datasets using TMT and TMT Pro. And then also we used MS2 and MS3 quantification. In the mix, there's also 2 non-ILQ datasets. So what we can see here on the right-hand side is at the top, we have the identified protein groups, and at the bottom, we have the identified precursors. The blue line shows us the performance of identifications of SpectroMine, and the gray bars show the other competitors. And what we can nicely see here is that SpectroMine outperforms all of the other softwares. Now the question is, what does that mean in terms of processing speed? And also here I can show you that SpectroMine outperforms the others. Let me quickly tell you what the plot is about, because it's a bit counterintuitive. So we're not plotting the processing time where you would assume the lower the better, but we plot here 1 divided by the processing time. So the higher the better. Where we can see here in the blue line, again, the performance of SpectroMine, and in the gray bars, the performance of the other softwares. So since I also want to highlight a more complex data set and also the comparison of SpectroMine 2 to SpectroMine 3, we have here now an explicit example. This is from a publication looking at a TMT 10-plex data set. It's phospho enriched and fractionated, and we can see in light blue SpectroMine 2 and in dark blue SpectroMine 3 performance. On the left-hand side, we have the identified precursors, and on the right-hand side, we have the speed. So we can nicely see that SpectroMine 3 not only outperforms competitors, but also that SpectroMine 3 in comparison to SpectroMine 2 got a lot faster while still maintaining the amount of identified precursors. And when we look at the quantification accuracy, we have a data set of a human steady sample where there's yeast spiked in at 3 different levels. So at 3-fold, 2-fold, and 1.5-fold. It's a TMT 11-plex experiment with 11 fractions and MS3 quantification. And what we can see on the right-hand side are the expected values here in dashed lines, and that all the quantification values nicely peak around these expected values. And with that, we are already at the part where we can look at the software and not just see the performance. And for that, I have a third polling question for you, where I would like to know how important is it for you to browse raw data actually? So yes, you use raw data visualization regularly, or yes, you think it's important, but you don't use it very often. Yes, but the tool that you're currently using does not have the functionality in the way that you like it, or no, it's not important to you at all. And I think we can already have a look at the results. Okay, so that's quite nice to hear that you, as well as we, think visualization is quite important. And then you're also in for a treat on that part. So in SpectroMine, we have 8 different perspectives or tabs. We have the archive perspective, where you can look at search archives or spectral libraries, basically at experiments that you've already created. Then we have the analysis perspective, where you can set up your experiment. You can review your runs at identifications from protein to PSM level. At the post-analysis, you can look at summarized information, like plots over the whole experiment, some hierarchical clustering. In the report perspective, you can export reports, but you can also customize your report setup to the way that you want it and even rename columns. In the QC perspective, you can monitor the performance like chromatography. In the database perspective, you can import or export like your protein databases, modification databases, and many more. The settings perspective, as it says, gives you the different settings like the search settings, experiment settings, or global settings. And in the about perspective, you can look at the software version or get a link to the manual. So today we focus on the 3 perspectives: the analysis, the post-analysis, and the report perspective. Also there, most of the changes happened. So in the analysis perspective, we have a fast track buttons now where you can save or refresh a post-analysis, but still for the users of SpectroMine 2, the right-clicking option is still available. Then up here, we can select what we want to have a look at. So for instance, here it's the PSM plot. But one thing I want to highlight is the analysis log, since it now has the option to filter. So you don't have to scroll through it. You can instantly filter for warnings or errors. Then if one plot is not enough, which quite often happens, you can additionally pop out an additional window where you can plot something else. And then also newly, as indicated here in the PSM plot, we have all the different fragment ions, we have the reporter ions, but also now new, we have the diagnostic peak as it is defined in your modification. And last but not least, sometimes you would like to look at various results next to each other. So we have the option to detach perspectives and then reattach them again. Let's have a look at the analysis perspective. Here you have a multitude of plots, and the time is just too short today to show you everything. So we have experiment overview, run overview, we have scoring histograms, but I want to highlight the two regulation analysis nodes. So we have the PTM regulation analysis, which is a new node, and we also have the differential abundance. And these two nodes give you also a multitude of plots where I just want to highlight a couple of them. So we have, for instance, the candidates plot, where you can look at over underrepresented candidates. We also have the option to look at the p-value distribution. We can also do some hierarchical clustering or just a PCA analysis. The last perspective I want to show you today is the report perspective, and here we have now new reports. So we have the run report, and that was already available in SpectroMine 2, where you have the reportables on a run level. Then new, we have the reportables per summarized fraction in our summarized fraction report. And last but not least, we also have the PTM side report if you do a PTM analysis. Both can be looked at in a long report, the normal report, or also in a short report, a pivot report. And what I would highlight are now the different columns that you have for these different reports and the levels. So for the run report, we have from experiment level to fragment ion, basically on every level, you can have a different reportable, you can look at quantities, and you can look at other analyte information. Then we have the summarized fraction report. Here we have the level experiment, sample, protein group, and peptide. And the beauty here is that the fractions are summarized. So everything that is put into a block during the experiment setup is summarized into a sample. And last but not least, we also have the PTM side report, where we have on experiment, run level, and the protein group, all the PTM sites that were localized. But if you do a fractionated experiment, then the run level will become the sample level. So it's also summarized across fractions. Something that I really like about SpectroMine is also that you have command line, because quite often, especially if you have a multitude of experiments, it can be quite tedious to click all the time through the GUI. And for there, we also have the command line accessibility. So if you want to know the specific arguments, you can have a look at the manual, which is also linked in our software. Or after you've set up one experiment in the GUI, there's the neat way to just export everything as a batch file. So there you have all the settings. So search settings and experiment settings are exported, and all the arguments are summarized in a text file. So you can also have a look there and learn from there. So to conclude this, SpectroMine 3 gives you the possibility to effortless set up Isobaric labeling quantification. It's super user-friendly and includes the latest TMT tags up to TMT Pro 18. It's dramatically faster, also to the previous version, but also in contrast to competitors due to our fragment ion indexing. Then it's the most powerful search engine, so you can quickly get unsurpassed protein coverage. We support ion mobility, so today I did not have the chance to show you that. We support library generation, and as you've seen, there is a multitude of visualization available. But for those of you who want to know more, and since 20 minutes are just really not enough, we have 2 videos by my colleague Fabia Simona. So one is how to jumpstart your data analysis. So this is more for the beginners, but then also what is super interesting is looking at what is new in SpectroMine 3, where you can have a look at an example of a fractionated PTM-TMT dataset. And with that, I want to thank everyone at Biognosys, especially the R&D team. Without them, the software would not be what it is today. Thank you very much. And I give back to Max.
Thank you very much, Takira, for this very nice overview of SpectroMine 3. And at this point, I would like to introduce our guest speaker, Erwin Schow from the Technical University of Denmark. He's been an associate professor at the Department of Biotechnology and Biomedicine since last year and works on hematopoiesis. So in order to really deep dive into cell differentiation, he develops cutting-edge single-cell proteomics workflows. And today he will share with us some of his work of using SpectroMine to quantify proteins in single cells. One note before I hand over, though, you can ask questions in the Q&A box. I see a few of you have already done that. So we will have a Q&A session at the end where we answer questions. So if you have any questions for Takira or for Erwin, just put them into the Q&A box and we will get back to you. Yeah, but with this, I would like to hand over to Erwin. And yeah, thank you very much for being here and presenting at our seminar.
Thanks a lot, Maximilian, also for that kind introduction, and of course for having me here. I'm very excited to kind of give you guys a brief overview of what we've done using SpectroMine in our single cell work and how we're kind of trying to push the maximum performance out using both advanced data acquisition, but especially data analytics for improving our single cell proteomics experiments. So today I'll be talking a little bit about the challenges of doing low input and single cell proteomics and kind of explain how we've established such a workflow, and then spend the vast majority of my talk on how we can use advanced data analytics as provided by SpectroMine for trying to get the deepest proteome coverage possible. So, I mean, most of people here at HUPO will be very aware of this, so I can go quite quickly through this, but really it's been this transition of going from where mass spec-based proteomics needed a lot of material, several microgram, if not more, for PTMs, and that these conventional sample preparation workflows are subject to significant sample loss. I mean, given all the steps involved with, potential for sample losses along the way, for having to optimize for single cell, we have to minimize all the sample loss and maximize the reaction kinetics by downscaling in volume, and I'll get back to that. But basically, just to give you guys an idea of the complexity of this, I mean, a single mammalian cell only has about 250 picogram, whereas yeast and bacteria are really pushing the envelope of sensitivity with 5 picogram or even less than a picogram. So I think there we are still, some time away from being able to do that. But indeed, significant efforts by us and many others have really optimized the sample preparation in order to be able to do single cell proteomics. And I think this workflow of taking your tissue and perhaps sorting for cell populations of interest, denaturing your proteins, cutting them into peptides, separating those peptides and introducing them to the mass spec are all good challenges that can be improved in order to improve single cell proteomics. So we really like to classify them as the 3 pillars of optimizing single cell, where we go from sample prep to sample separation and ionization, and then of course also instrument methods, but very importantly also once you have the raw files to maximize what you can extract from this data. I mean, I don't have much time today to talk about how we attacked or are trying to tackle all these different aspects, but there's been some really nice editorials about this in Nature Methods, and Chemical and Engineering, where a lot of the work from key researchers in the field has been highlighted. So I would highly recommend to read those if you want to know more, and also these references listed at the bottom where you can see what's possible and what's been done. So how do we go down to single cell proteomics? I mean, we were really inspired by the SCOPE-MS approach from Bogdan Budnik and Nikolai Slavov, because indeed we have this challenge of getting enough peptide copies. And as I showed before, 250 picogram is really very little. And to overcome the fact that we have so little material, we instead opted to use this booster or carrier channel, where we can provide extra peptides and consequently more ions for identification, and to use TMT Pro to multiplex these single cells with this booster channel and get the cell-specific quantification. So you've already heard a lot about TMT Pro from Takira, so I don't need to go in too much detail, but I mean, really indeed just wanted to stress that we label all these single cells with unique tags. We label the booster channel with unique tags. And because of the isobaric nature of this labeling technique, it means that the intensity at the MS1 level gets summed into one more intense peak consisting of all these single cells plus booster. And then as TMT Pro functions, we have to fragment these peptides into MS2 spectra, where we get our identification peaks for the peptide ID and then get these nice clusters of reporter ions to quantify the peptides of interest, and especially then at the single cell level. And again, this is now widely used and provides both an improvement in terms of throughput. We can run 16 single cells per injection, and we argue that it also has great sensitivity benefits. So how do we prepare these cells? So we were very keen on using FACS sorting so that most research institutes who have such a machine can use the workflow, while indeed also be able to record the FACS markers that you might be interested in. I mean, in the field of hematopoiesis or immunology, we are quite capable of finding specific cell populations based on their surface markers. So we can record this information as an index sort while depositing the cells into a 384 well plate. And in each well, we first deposit 1 microliter of lysis buffer based on trifluoroethanol. So it's all volatile salts that can just get speed vacked out. We don't need to do cleanup. So it's a really nice and easy workflow. So once we deposit the cells, we basically freeze the plate. We boil the plate at 95 degrees for 5 minutes, and then use a non-contact liquid dispenser to first dispense the trypsin for digestion, do an overnight digest, and then use this liquid dispenser again for labeling the next day. And why we use this liquid dispenser is especially for the randomized TMT labeling, where we try to basically randomize across our TMT pools which cells get which labels, and trying to pipette this by hand is not very recommendable. Then finally, I mean, we can do this by hand with an 8-channel pipette, but we decided to let a robot do that work for us, the OT2 from Opentrons. And here you can see the single cell plate. Here's the booster channel plate. So we just prime the tips, and then we start transferring over all the single cells back into those wells. And this plate gets speed vacked and then resuspended and put in the mass spec for analysis. And all this work has recently been published in case you want to read more about this. So at the beginning of these experiments, we weren't quite sure how to analyze this and whether we could extract biological meaning from this. So we basically decided to run 2 384 well plates at 2 different mass spec settings. So one using 300 millisecond injection time and the other using even higher of 500 milliseconds. To then ask, okay, if we sort LSC, progenitor, and blast, can we afterwards, using the proteomics data, separate out these cell populations? I mean, in terms of IDs, we get about 1,000 proteins per cell in the 300 millisecond and about 700 proteins per cell in the high data set. And if we then visualize this data, so these UMAPs are drawn on those proteins we identified, we can see that especially in the 500 millisecond, we're separating out these blasts in blue very nicely from the LSC and progenitors. So this is a leukemia culture system consisting of multiple cell types. It's from a primary AML patient. And we know that these LSC and progenitors are very different, so we should be able to, from the blast, so we should be able to separate them. And we can see that in both cases we can do that, but that especially in the case of the 500 millisecond, we can separate out these progenitors from the LSCs, which we're not so well capable of in the 300 milliseconds. So that's quantified in this plot, kind of showing that we are better able to separate progenitors and LSCs using this very high injection time of 500 milliseconds. Then compared to bulk proteomics, just to see what the accuracy is like, we can see that we have quite decent Pearson correlation between single cell intensities and bulk proteomics intensities. And if we narrow that down to the top 400 proteins, and we are actually very good at quantifying these proteins. So how can we take this further? I mean, of course, 1,000 proteins per cell is nice, but we would like more. And I think everybody in the field is very keen on pushing this further. So we thought to work with Biognosys on figuring out how we can improve this. I mean, we have to all work together to make single cell proteomics a mainstream technology. And I mean, we want better cell throughput. I mean, we are doing about 140 cells per day per instrument, but of course, we'd like to make them more like 300 cells so that a 10,000 cell experiment would, quote unquote, only take 1 month to run. So we need shorter gradients, we need faster instruments at the same level of sensitivity. Of course, higher multiplexing would help, but also removal of the booster channel to free up these 2 extra channels. At the same time, and I know these two are kind of counteracting each other, we also want deeper proteome coverage, you know, ideally somewhere between 3,000 and 5,000 proteins per cell. And I think for this, we're really going to need smarter data acquisition. We've seen great effects of better sample prep, like the nanopods or the proteo chip. We would really like something that has full fact sorting compatibility. And of course, here also more sensitive instruments, better chromatography will help. And then the data analytics will also go a long way. And today I'll, of course, be talking about these data analytics, but I also would like to kind of show our evaluations of trying to remove the booster channel and what effect shorter gradients can have. So, I mean, we really were excited about using SpectroMine because of this library search that Takira so nicely explained about. So we first started with a dilution series. So it's a bulk 12-plex TMT Pro sample diluted to 125 picogram per channel. I mean, it is a dilution series. So if we dilute it to 250 picogram, we were really overloading the system compared to real single cells. So we, per injection, have 1.5 nanogram total load in triplicates. And then the booster is added into 126 throughout this presentation. And the experiments were done using an EvoSep with whisper flow on an Eclipse run in MS2 mode, just to simplify kind of the experimental readouts. So if we vary the amount of boosting we do, so going from no booster to 25, 50, 100, or 200 cells, you can see that, first of all, on this dilution series, we're not seeing much of an effect of increasing the amount of boosting. Again, suggesting that dilution is not quite reflecting true single cell. So here we're running 10 SPD, which means 10 samples per day or a 2-hour gradient. And if we run the standard DDA mode in SpectroMine, we get these consistently above 1,000 proteins, even up to 1,200 proteins. If we then use these results and construct a spectral library from all those samples and then research, we can see that we get some benefits in terms of increasing our identifications. And we love all benefits we can get, especially if they are for free by just reusing your raw files. And you can see that indeed we are looking at a few hundred more peptides per analysis. If we then shorten those runs to the 20 SPD method, i.e. 20 samples per day or 1-hour gradient, you can see that with the higher boosting, we are still well above the 1,000 proteins in the single cell channels. But indeed, we are definitely identifying less. And then it's really up to the individual researcher to decide what they find more important, but still getting quite good numbers of peptides. But dilution series is one thing. We, of course, want to do this on real single cell. So we repeated this experiment basically now using 18 real single cells prepped according to the workflow I presented, and adding booster into the 126, i.e. masking that single cell that's there in these booster samples. So again, we're checking just with standard DDA and seeing a nice increase in numbers of proteins identified in the single cell channels when we use real single cells. If we then again make the spectral library, we get a small bump in terms of protein numbers. We're getting a slightly larger bump in terms of peptides, like 100 more peptides, which is again 100 more quant values, meaning it's a welcome improvement to all this. And already without the library, this was about 5 to 10% better than competitive software. So we were very pleased. This is run with 120K res in the orbit trap and then 500 millisecond injection time. And then if we decrease the runtime to 20 SPD, we are, again, like in the 10 SPD, able to identify about 500 proteins without any boosting, which we actually were quite pleased with. We hadn't seen such performance before. And then with the 20 SPD, we do run a shorter injection time because the peaks are sharper. So we have more intense ion flux, meaning we are allowed or we are able to inject shorter and therefore have a higher scan speed. But still never quite get to the 1,000 proteins per cell in this case. But still, I would argue that for the trade-off of cell throughput, this is still most likely going to be biologically informative. What's also interesting is that indeed, especially on these low amounts of boosting, we are getting a significant effect by adding this library search. I mean, more than 10% improvement in terms of proteins and peptides on these short gradients. We're seeing about a 3% increase on these longer gradients, so slightly less than the short gradients. And you can see that as boosting increases, your benefits decrease, but this is exactly also the point that we would like to improve these low amounts of boosting through library searching. So people in the field are very correctly looking at signal to noise, because of course we are trying to quantify very low amounts of signal. So I just wanted to show that, with these two different gradients and two different instrument methods, we are seeing different types or different levels of signal to noise. So I mean, booster free on the 10 SPD with the long injection time really gives a lot of signal to noise. So this is median signal to noise in the single cell channels of 41 compared to 24 in the 20 SPD. And we were quite pleased to see that even after adding some boosting, this didn't decrease much on the shorter gradients, the 20 SPD. Of course, it did decrease on the 10 SPD, but you are starting to identify more, and therefore probably the amount of proteins that you can accurately quantify will have increased at this point. And then once you go up to these levels of boosting, you're not seeing much difference between these methods, meaning that you probably don't need to worry too much about the boosting. But yes, we are indeed confirming what was shown by Christopher Rose's lab, that the more boosting you add, the less signal to noise you can expect from your single cell channel. So it's something you need to evaluate very carefully in each and every experiment. So then we wanted to see if we can kind of optimize this library search. So we, as I showed before, we use a Fasta search, we construct a spectral library from that. So what if we run the same sample on 10 SPD, 2-hour gradient, but then decrease this injection time to try and increase the scan speed, but not caring about the single cell quant? This is just to get more peptides into the library. So you can see the comparison between the long injection time and the 246 millisecond injection time, that we are indeed identifying significantly more proteins, especially in the higher boosting, and also at peptide level, we're seeing many more IDs. So we then thought, can we, we also, just to show that the single cell signal to noise is really worse at these low injection times. So I wouldn't recommend this for the actual single cell quant, but for building a spectral library and more like a hybrid library, we just wanted to explore whether this could boost our identification rate even more. And luckily, this was indeed the case. So you can see here the DDA on this 10 SPD data set, the first library and then adding this Fasta scan speed library, where we suddenly start adding like 30 proteins without any boosting. And in terms of peptides, again, are seeing about 100 peptides more. And again, this is just costing you a few extra runs, but then you get the benefits for all the single cell samples that you will run afterwards. So for sure, you can add more IDs by running your machine in a different way. Then the last part of this library analysis, we wanted to see what happens if we use different libraries for the respective samples. So we're checking here, for example, on the booster-free samples, what happens if we use the booster-free library, the 25-cell library, or the 50-cell library, the 100-cell or 200-cell library. You can see that the 200-cell library doesn't actually perform any better, and maybe even a little bit worse than the booster-free library. And then with the 50-cell, we start seeing that the no-boost library performs worse than the 50-cell, but then we don't see an increase going further up. And the same at the 100-cell, that seems that the 100-cell booster is performing best, and at the 200-cell samples, also the 200-cell booster library performs best. And the pattern is very similar at the peptide level. So this was quite interesting to us because it does suggest that sample load specific library searching performs best and is consistent across the entire range of boosting. I mean, we came into this kind of hoping that the 200-cell library would improve a lot the booster-free setup, but that doesn't seem to be the case when we do such a focused analysis. But I mean, this is not quite a fair comparison because we show that if we pool all these libraries and then search the no boost, we do actually get this benefit of 50 to 100 proteins and more than 100 peptides. So this hybrid library is definitely something that outperforms DDA and does still provide a fruitful strategy where you just pool everything. And these results here are based on only 4 runs, whereas the 16 runs compiled into this hybrid library showed that there was actually a real benefit. So just to get back to our wish list, I just wanted to spend the last 2 minutes or so on trying to showcase how we can also use smarter data acquisition combined with this advanced data analytics. And for this experiment, we tried to utilize the fact that our Eclipse tribrid system doesn't only have an Orbitrap, which we need for the full 18-plex reporter ion resolving. I mean, we're talking about 0.01 Dalton mass difference, so you need this ultra high resolution. But it also has a linear ion trap. And the linear ion trap, it's a lower resolution mass analyzer, but it has higher sensitivity. So we were kind of curious to see whether this could be an alternative strategy. The main disadvantage of this is that the lower resolution means we need to decrease our multiplexing capacity to the 1 Dalton different TMT tags, meaning we have to restrict to 10 cells or 9 cells plus booster, because we can only use either the N or the C variants of TMT Pro. But what we saw using this got us actually extremely excited. I mean, if we again take this DDA approach from SpectroMine and use the 1-hour gradient, no boost seemed to underperform compared to Orbitrap, and we're still figuring out why that happened. We'll do lots more replicates, but with 50-cell boosting already, we are well over the 1,000 proteins. Now with 200-cell boosting, we're actually now starting to approach the 2,000 proteins per cell mark with 6,000 peptides, which we haven't seen before, that level of performance. If we then add the library, we're starting to really see some nice results, and I can quantify those. That on the booster-free sample, we're suddenly having 35% more proteins, 42% more peptides. And then even with 50-cell boosting, we're still having more than 10% increase just by researching the same data using a library compiled of all these runs. And we are again seeing this phenotype that as booster increases, the benefits of library-based searching decrease, but still very much are worth doing. So we are really now starting to approach the 2,000 proteins per cell, which got us extremely excited. Because at 20 SPD, even with 9 cells, we would be outperforming our throughput of 140 cells and would be running 180 cells per day. But of course, there's a lot of work now. We need to evaluate reproducibility, the quantitative accuracy and precision. But this does seem to have some potential for improving our analyses. So just to conclude, I mean, SpectroMine really is very suitable for single cell mass spec raw file processing. It truly is a lot faster. I mean, sometimes I'm setting up a search across different files, and then the first one's already done by the time I'm done setting up the whole search. We have more proteins than competitive software that we've been using extensively in the lab. 18-plex was nice to see that was integrated out of the box. We didn't need to program any of that in. I hope I managed to show that library generation has to be optimized on a per case basis. So you're adjusting your sample load and instrument settings. We're ongoing to work together with Biognosys on trying our reticle approach that we have on bioRxiv and is currently under review, where we do a quick pre-scan in the ion trap before sending it to the Orbitrap for the long injection, high resolution scan that we hope to see some benefits. And seeing how well the spectral library was working on the linear ion trap, we're also very keen on exploring this SPS-MS3 co-isolation free analysis of single cells. So with that, I really want to thank a lot of people, of course, Biognosys, to Kira, to Jas and Lucas for this wonderful collaboration. Bo, my former postdoc supervisor, without whom we would not be doing any single cell proteomics. Benjamin and Neil are two PhD students that joined to do single cell proteomics. John, who is my other former postdoc mentor, Ulrich auf dem Keller at DTU Proteomics Core, and all our other industry partners, and of course funding. And with that, I would be happy to take questions.
Thank you very, very much, Erwin, for this great insight into your work. I want to first remind everyone that you can ask questions with the Q&A button. And I turned my video off, but I would also like to point out before we get to another polling question that SpectroMine is available as a free trial version where we do a guided trial and help you get started with the software. So you can try it out for 30 days for free. And with that in mind, would you like to try SpectroMine? And please answer in the polling question now, just so we get a bit of an idea of the level of interest in the room. Okay, I think we can close the poll here. And yeah, we have a lot of people interested in trying it out. So yeah, just get in touch with us. We have information available on our website, biognosys.com/spectromine-3. And yeah, now getting into the questions. So while you can still submit questions for Erwin. So maybe let's start with a question for Tekira. Let's see. How does SpectroMine deal with large TMT experiments using blocks?
So SpectroMine natively supports the concept of blocks. So you can, when you set up your experiment, create blocks and put the runs within a block together that are summarized, and you can also do a condition setup. And additionally, what you need when you have a blocking experiment, obviously you need a reference channel that you can specify, and this is then used to normalize across the different blocks to make the quantitative information comparable between these blocks.
Mm-hmm. All right, so I'll just switch over to a question for Erwin here. So this one has received an upvote. So what do you think of DDA for single-cell proteomics, or is multiplex DDA just inherently better for this use case?
That's a great question. I mean, I think they both have their own merits, and yeah, we'll probably see two camps continuing in the field on pushing both, and I think that's only fantastic. We need all the kind of evaluations that we can get. I mean, we pushed for multiplexing mainly for throughput reasons. I mean, with 16 cells at a time, even with the 20 SPD, we are looking at 360 cells per day, which would equal to 4 minutes of runtime per cell. I mean, doing DIA at that level, if we can do it, I probably would indeed switch. I'm not seeing that happening anytime soon. I think indeed what we're seeing so far is 40 cells per day, which indeed looks really good, and the performance is absolutely stellar. But if we want to kind of start competing with single-cell RNA-seq in terms of attracting real biological questions to this technique, we need to try and match these 10,000 plus cell experiments. And I think indeed running mass spec for a month to do that is fine, even probably a few months, because data analysis generally takes longer. But beyond that, I think, yeah, you're going to run out of time to do the biology. So this is mainly why we went with the multiplexing approach.
Mm-hmm. Thank you. So for Takira, here's someone asking, and this is probably because the talk was focused on highlighting the software in general, but someone's asking what the specifically new features are in SpectroMine 3 compared to the current version.
Yeah, that's a very good and valid question. So SpectroMine 3 in comparison to SpectroMine 2 is a lot faster in terms of processing immunopeptidomics or many modification searches. So I think that I showed, and we also improved our quantification settings. We improved impurity correction for TMT, default settings, turbo TMT. Then we also have now the 18-plex available. We've added improved support for fragmentation, so that you have this nice new report now, and also a node in the pub, as well as for PTM support. And I'm pretty sure I forgot things, but the good thing is that we have the release notes also on our launch page, where you can then check it out and just read again, like all the things that we've changed now in SpectroMine 3.
Thanks, Takira. So again, a question for Erwin here. Let's see. So yeah, someone's saying, nice work, Erwin. You mentioned the need for higher multiplexing. And what do you think about hyperplexing approaches? And do you think there will be a label available that goes beyond TM2E Pro 18-plex in the near future?
Nice. I like the question a lot, because indeed we can't unfortunately see in the future. Thermo's being rather quiet about any kind of beyond 18-plex, but we're all hopeful that within a year or two we might see a doubling of that, and probably then we start running into ion multiplexing issues that we can collect enough to spread across so many samples, but I think that should still be possible. Hyperplexing is of course very exciting if you have a model system that you can label with SciLac. So indeed, there's probably questions that can be asked to do this. However, you also got to then realize you will need to isolate and fragment 3 different groups of precursors to then get the quant of that 3 × 18 single cells. So your scan speed will undoubtedly suffer a bit, but absolutely, I think there will be opportunities to test that. But of course, we're looking a lot at primary patient material or healthy bone marrow donors, and there it's not an option. But yeah, absolutely, in the lab, there'll be questions that can be asked that way.
Yeah, for some cases, a good option. Okay,
yeah.
So yeah, another question for Takira. Actually, it's 2. So here someone says, thank you for your immensely great work on the software. When I generate and manage my ion libraries in SpectroMine 3, are they identical, PSM, etc., to ion libraries I generate with the same settings in the latest Spectronaut? Or is the search engine SpectroMine 3 more advanced than currently in Spectronaut? Yeah, so maybe you want to answer that one.
Thank you. So the good thing is that we just recently launched Spectronaut 15.5, and that version is identical to SpectroMine 3. So there is no advantage in using one over the other when it comes to library generation. So you can easily use both and it's compatible.
Yeah, and not to distract, right? But that also means that SpectroNaut has become a lot faster for searches with many modifications with this latest update that's now available to everyone who has the software. And then the other question here is, do you also support MaxLFQ and run alignment in SpectroMine 3?
True, that's something that I haven't mentioned. So we also have MaxLFQ implemented in SpectroMine. So it's under the quantification settings. It's per default switched off. We also amended the algorithm that it can be used for ILQ. However, in the TMT and ILQ experiments, we don't have that many missing values because, I mean, like the advantage of the algorithm is mainly tailored to LFQ. But yes, it's available and usable.
Thank you. And yeah, we unfortunately have to come to an end very soon. There's one question that I don't find right now, but it was about, oh yeah, have you tried performing these benchmark tests on peaks 10.6? It would be good to get a comparison on a more up-to-date version for all software products. So maybe I can just say a few words on that, because one of the figures that was in Tikira's talk was actually also comparing with peaks 10.6 and looking at the latest versions of all the other softwares as well. And for the comparison with more data sets, we looked at a few of them. There were no dramatic differences. I think for peaks, there is a slightly higher number of proteins identified now. But comparing protein numbers is a little tricky. So yeah, and when you look at the precursor comparisons, from what we can tell, there wasn't a dramatic difference there. Okay, and with that, I think we should close it because we're already a little bit over time. But yeah, I would like to thank Erwin and Tikira for their great presentations, and everyone for coming here and joining us for the seminar. If you have any open questions, you can always contact us. You can go to our website, biognosys.com/HUPO2021 or biognosys.com/SpectroMine-3, and there you can find all the information about our software. So thank you very much, and see you for our next seminar in the future. Goodbye.
Thank you.
Thank you. Goodbye.